Muscle contraction happens when actin and myosin filaments slide past each other, shortening the sarcomere. The axon of the nerve cells of the spinal cord branch and attach to each muscle fiber, forming a neuromuscular junction. Nerves release calcium. Acetylcholine is then released. Acetylcholine binds with receptors and opens sodium channels. Sodium rushes into muscle cells. Muscle action potential sweeps into T-tubules. The sarcoplasmic reticulum releases calcium. Here you see myosin, which has ADP and phosphate at resting stage. It also has actin, troponin, and tropomyosin. These molecules bind with troponin. This causes the tropomyosin to move, revealing myosin attachment points. The single phosphate on the myosin head is released as the head moves to connect with the attachment site on the actin. This formation is called a cross bridge. The remaining ADH is then expended into what is called a power stroke, when the myosin heads pull the actin inward. Once ADH is spent, ATP attaches themselves to the myosin heads. This molecule triggers the release of the myosin heads from the actin attachment sites and are immediately broken down into ADP and a single phosphate again. Relaxation occurs when stimulation of the nerve stops. Calcium is then pumped back into the sarcoplasmic reticulum, breaking the link between actin and myosin. Actin and myosin return to their unbound state, causing the muscle to relax. Immediately after death, the muscles of the body contract in the same manner as they do when the person is alive. When breathing and circulation stop, muscle cells lack oxygen and therefore cannot use aerobic respiration to efficiently produce ATP. Respiration continues at first, but the muscle cell eventually becomes so short on ATP that the myosin and actin filaments cannot release from the contraction state. The calcium ions cannot be pumped back into the muscle cell. Unable to release contraction, muscles remain tense.